Good morning, Children's Health Defense. I'm Mary Holland, CEO of CHD, and it is my great pleasure and honor to have with me here today Dr. Anna Michalczak, who is both an MD and PhD, and she's going to be talking to us about her study of blood in both vaccinated and unvaccinated people who've taken with COVID shots. So Anna, welcome.
Thank you so much for having me.
So first, I'd like for you to tell us a little bit about your professional background. What brought you to this? What has your sort of study journey been like?
So I am an internal medicine physician, board certified. I have a PhD in pathology, and I have about 25 years now of clinical experience. I used to be a geriatrician, and the reason why I got into all of this was because once the pandemic started, I got involved with different physicians groups like Medical Doctors for COVID Ethics International, and there I met some researchers, and specifically Dr. David Nixon, Shimon Yanovitz. And one day he asked me to look at the COVID vials under the microscope, and what I saw shocked me because I saw self-assembling nanotechnology. And after that, I got myself a microscope, and I've been studying and documenting and researching ever since, because I started seeing abnormal blood findings and self-assembling nanotechnology in unvaccinated blood from shedding, and the same rubbery clot formation we were seeing in the vaccinated as well. I analyzed some of the clots, so it's a big picture because it was all so tied into the geoengineering aspect, the chemicals from that. I work with Clifford Carnicom from the Carnicom Institute, who for 30 years did research on morgellons, and we found that there was an overlap between what was growing in the blood and this morgellon synthetic biology.
Okay, so let's go slowly though, because a lot of this may be new for viewers. So you've been studying blood using dark field microscopy, is that right? What is that? Explain that to us if you would.
It is a type of a microscope that's extremely sensitive, so it goes up to 4,000 times magnification, and it uses dark field rather than bright field, so you can see extremely small things with much more contrast.
Okay. And so when you started looking at what was in the vials, and then you started looking at blood samples, tell us a little bit more about what you saw or what you see. And I guess the image behind you is related.
Well, how about I show you, because I just recently did another analysis on the COVID vials. So what you see here is a Pfizer BioNTech COVID-19 injection when it was first put on a slide. And in the background, you see all of these blinking lights and extreme activity. This is 100 times magnification, but this is exactly what I saw the first time 3 years ago when I looked at it with Dr. David Nixon. And so this turns out to be these quantum dots, nano and micro robots. And here you see that something is self-assembled that we call microchips, because we have done studies that these microchips are actually emitting what's called a MAC address phenomenon. And so this is sort of an initial view of the vial. Now, if we go to a larger...
One second, Anna, what are the tell us what the flashing dots are in the background.
So this is what's called nano and micro robots. They are communicating with each other via light signal, and they are collaborating to self-assemble these larger structures that are microchips. And so this is in the realm of self-assembling nanotechnology. Here you see a micro robot emitting blue light. Here's another one. And basically these little tiny robots are coordinating and are then growing larger structures and building them. Here you see different images of what is being self-assembled, which other colleagues around the world have also shown. And these are these complex structures that grow within the vials. And what is very important is that this background swarming is what I've seen in the human blood, and I will show you later images of that. And what is important is that in the engineering world of nanotechnology, the head of the IEEE, or a very prominent professor, Dr. Ian Kildes, discusses the Internet of Bio-Nano Things, and he basically explains how nanomaterials such as graphene have inspired the idea of connecting biology with synthetic biology and then into the engineer of biological embedded computing devices. And so this is then what creates an intra-body area network that is used for biosensing. This is part of what we now know as, for example, the brain-computer interface, as well as in medical applications. For example, there are certain drugs that contain microelectronics and have been studied. For example, Pfizer's Enbrel, which I've also done analysis on, and they have these microelectronics in them that self-assemble in the patient's body.
And were these in Enbrel, let's say Pfizer's Enbrel, were these nanotechnology devices, were they disclosed in their patent and in their product insert?
In the Enbrel case, there was a study done by Mount Sinai Hospital that specifically discussed the microelectronics in Enbrel, and then how the doctors were collecting the biophysiologic data to the patient. But most people do not know. Obviously, with the COVID shots, this wasn't disclosed.
Okay.
And here you can see exactly how it works. So you have a artificial cell that has inside of it nanotransmitters and biological nanosensors. And this is an engineered cell. So this field is very confusing to doctors because they were not trained in nanotechnology or electronics to be able to discern this. So we have to really have a cross-domain and many scientific disciplines discussion to be able to find the answers to this. This was a summary that was published by Dr. David Hughes, who really looked at the 26 teams worldwide. In the meantime, there have been a lot more who looked at the vial content and found that, the RNA really was not conforming to a label. We know that DNA and protein impurities are in there and that many metals were found. And so I'm a chelation specialist and I specialize in detoxifying heavy metals. So these metals are part of self-assembling nanotechnology and semiconductor technology. And in the military battle space, they are used for battlefield interrogation. So the more metals an individual has in them, the better they can actually be surveilled via facial recognition technology. So this right here is from the university, the National Scientific and Technical Research Council, National University of Como in Argentina. Dr. Marcela Sangorin and biotechnologist Lorena Di Blasi, they did analysis of COVID-19 injection and found 54 undeclared chemical elements, including fluorescent graphene oxide. And I have documented the fluorescence phenomena in the blood and with this technology extensively. So specifically, they found what's called lanthanides. Lanthanides are neodymium magnets, for example, which is the most powerful magnetic substance on Earth. Praseodymium, hafnium. So these elements are used for semiconductor and quantum dot self-assembling nanotechnologies. And for example, silicone is used for semiconducting technology, and they have used polymers in them, polymer plastics that require aluminum and titanium as a start signal to self-assemble. So these metals have a very specific purpose. They are not just contaminations for no reason. And you mentioned that, Antonetta, Sagatti when we discussed, because I've looked at many childhood vaccinations now under the microscope, and they have the same self-assembling nanotechnology, and she has proven that the childhood vaccines also contain all of these numerous metals. So this is part of the technocratic transhumanist agenda that has been explained by thought leaders like Google engineer Ray Kurzweil, who want to fuse humans with machines. And this right here is a new scientific paper from colleagues from South Korea. Dr. Youngmin Lee analyzed 54 COVID vaccine vials, 45 Pfizer, 7 Moderna, and AstraZeneca and Novavax, and found exactly the self-assembling nanotechnology that I'm also showing, the same thing. She also incubated these over more than 2 years and showed that the self-assembly never stops. She showed also, for example, in semen samples of vaccinated individual that this stuff continues to shed. So we've done analysis on saliva, on sweat, on vaginal fluids, on peripheral blood. And this is emitted from individuals, which is also in the Pfizer documents that clearly states that if an unvaccinated man inhales the air around a vaccinated woman, that man can transfer the vaccine to another unvaccinated woman. And that's exactly what I've been documenting in the blood. So this is now unvaccinated blood. This is what we started to see, these polymer filaments that have very much clinical similarity to Morgellons that has been documented via geoengineering.
Tell us what is Morgellons. Not everyone's familiar with that. And isn't that a controversial medical diagnosis? Tell us about that.
So it's not a controversial medical diagnosis in the sense that there have been illegal geoengineering operations that have sprayed polymers with metals, including aluminum and barium, for 30 years, and that these synthetic biological organisms that have been isolated by people like Clifford Carnicom, they were called Clostridium bacteria, they have been cultured, and they're synthetic biology, and people have been infected with that and marginalized as delusional parasitosis. But it turns out that we chemically analyzed the blood samples and the environmental filaments, and they have a match, meaning we found through near-infrared spectroscopy now in vaccinated and unvaccinated blood chemical signatures of polyvinyl alcohol, polyamides, and polyethylenes. Those are unnatural microplastic signatures. These are part of this entire geoengineering program.
So this is a separate avenue than the vaccines, or you think that these filaments are also in the vials?
So the same vials that were found from RAIN analysis chemically have an overlap with the filaments that we're now finding at these rubbery clots. There is a chemical match going on. And how I really figured out how this self-assembled was that Richard Hirschman sent me embalmed blood of a vaccinated individual who had been deceased for 8 months before. I got these vials in June of 2023, and you can see here the same filaments. And this is a video of ongoing self-assembly within this embalmed blood. And what you see here are these micelles, and they're filled with the same blinking lights that I showed you earlier in the COVID shots. This is micellar self-replication, and it is ongoing. And a little bit later, I'll show you, I re-looked at it a year later, just a couple of weeks ago, and it continues to self-assemble.
What would 8-month-old blood typically look like, Anna? What would you expect to see?
Well, first of all, I would never, ever expect to see blinking lights in human blood. This is highly abnormal. You see that they're emitting light in different colors. This is 4,000 times magnification. None of this is biologic. It doesn't exist in the human body, And so this is microrobotics. But once I saw it, then I looked at my patients with unvaccinated blood, and I found these same structures with the same nanoparticles, and I filmed how they're actually building the filaments. You can see here that they are together, fusing together to build this filamental structure. And this is all part of the self-assembly. These spheres, they get rid of their contents to build these filaments. And so that then contributes to the clotting of the blood. Here is this same embalmed blood in September 24. So it's been now over 2 years that this individual is dead. This is 2,000 times magnification. And if you look at this bubble right here, number one, there's all these nanodots emitting blue and red and yellow light. And you can see how this sphere is literally now continuously growing. I'm speeding this up so you can see it a little bit better. And there it is. So this is happening in embalmed blood, ongoing. And this correlates with the fact that researchers like my colleague, Dr. Pedro Chavez from Mexico, has found ongoing MAC address emission from grave sites of injected individual. This technology doesn't die. It continues to emit. This is 4,000 times magnification. You can see dots here that are maybe 500 nanometers in diameter. I can see to the nanoscale with this microscope, and you can see that it's moving and it's still blinking. This is definitely still present. If you see here, this is the embalmed blood, and this right here is a micro robot. Okay, so look at this blinking light. You see that the cells are dead, and this thing is able to maneuver around the cells and, continue to travel. It's clearly emitting light, which isn't something that is normal and shouldn't be in the blood. And so that is still going on even after death. Here you see also embalmed blood, and all of these blinking lights here. You see these robots, they're able to maneuver extremely well surrounding these blood cells. So, and they're emitting light. That's a robot right there.
We think of robots as being controlled by some external force. Do you think there's something external controlling these robots, or are they self-programmed so that they're really intelligent and finding their way among the blood cells?
Yeah, so they have swarm intelligence, but they have biosensors in them, which are used for what's called bidirectional telemetry. That means they send signals to the cloud via cell phone and then for biometric data, but they also can be programmed externally. So if you see, for example, here, this is a robot. How do you know it's emitting blue light? Nothing in the cell, in the body is supposed to blink blue, okay? And you can see that it's very intelligent, and these are coordinating in a swarm-like fashion to build different things that I filmed, in human blood. This right here is what's called a mesogen. So I work with Dr. Hildegard Stanning, a world-renowned toxicologist who was investigating brain chips from targeted individuals and written books about it in 2009. And it turns out that this right here is actually a mesogen chip, and all of these little nanoparticles are self-assembling it. So what they're building is electronic circuitry. And I filmed this live in unvaccinated blood now, and people who've been exposed to shedding. Here you see 4,000 times magnification. You see the same nanoparticles that I showed you in the Pfizer vial. None of this is supposed to be in the body, blinking, emitting lights, and intelligently, coordinated. So this is highly abnormal. It's not biological. This is technology.
Right.
This right here is a research article that discusses about mice mesogen microchip devices, which is here photographed in unvaccinated blood. So these are liquid crystals, and they're using polymers, which are these hydrogels, like the polyethylene glycol from the lipid nanoparticles is a hydrogel, which is used for brain-computer interface, and basically they can replace any cell, any part of the body, and build these soft robotics inside of the body. Here is what I want to show you about the rubbery clot. So we did a lot of experiments because this is vaccinated blood, 30 cc's just drawn, and you leave it to sit for 4 hours. And normal blood will create, the red blood cells here at the bottom, but then you see here is a rubbery clot-like formation. And this stuff is exactly the same stuff that the embalmers have found in the deceased bodies. And it turns out that polymers, when you lower the temperature, they start self-assembling and coagulating. I analyzed these clots microscopically and found these nanotechnological filaments in there. And Clifford Carnicom and we also did chemical analysis on them. I also filmed how the clots actually assemble. What you see here is unvaccinated blood. This is the nanorobotic swarm. You see all these green and orange and yellow lights, and they are hijacking the electricity from the red blood cells, and they're killing them basically to feed themselves. This is called energy harvesting. It's well described in the nanotechnological literature. And so these robots, then, what they leave in their wake is this. This is the same blood 30 minutes later. The blood is completely destroyed. The robots have moved on, and this is the rubbery clot that we've been seeing, and that you can see how it self-assembles and literally builds when you are leaving when you're drawing the blood and you are leaving it out for at least 4 hours. And that's an easy test to do. Any doctor who is maybe thinking, this is too far out can replicate it. It's very easy to do.
Yeah.
This right here is our analysis of the clots. Clifford Carnicom and we got the deceased clots from Richard Hirschman. We got a clot from a living vaccine-injured person and from an unvaccinated individual. And what we looked at is clearly this rubber-like material that we know about. And we did microscopy, and we found the same filaments that we see in the blood, absolutely the same stuff. Okay, and then we did near-infrared spectroscopy and found that these things have polyenes, which is polyethylene alcohol, polyvinyl alcohol, which is plastic, and polyamide, which is silk, nylon, or Kevlar. And specifically, Dr. Kernighan found genes of DNA of spinoidins, of spider silk. That's the strongest military-grade material that they have made. And so we found these in the clots. And Clifford also did analysis. We tried to dissolve this stuff, and we used extremely caustic agents like strong sulfuric acid, lye, acetone, alcohol, toluene, stripper, petrol, DEET, fuel injector cleaner. All solubility tests failed. So once it's self-assembled, you cannot dissolve it. You have to dissolve it, you have to dissolve the building blocks. And what I want to show here, this is the Moderna patent, okay? And it says lipid nanoparticle composition and the methods of formulating the same. It has a section that shows stealth nanoparticles, and they are made of polyethylene, polyamides, which is exactly what we found, polyvinyl alcohol, which is plastic. And by the way, they have, for example, polymetacrylates in there, which is super glue. Okay, so there are nanoparticles in there that have the same chemical overlap to the polymers that are also being sprayed via geoengineering. Here's some testing on therapeutics. Because I drew unvaccinated blood, you see the same rubbery clot here. If you use EDTA, which is a metal chelator that inhibits the polymerization, the clot will not develop. Also vitamin C, it will not develop. I've been using this therapeutically now for 3 years, very, very successfully. And guess what? Here's the same patent that shows nanoparticle inhibition. If you want to stop the nanoparticles from self-assembling, you need to use a chelating agent and a reducing agent. And what are they mentioning in the patents? EDTA and ascorbic acid. This is where I got it from, and it works.
Wow.
And so here I'm showing you a test that I just did this Monday. Here you see a Pfizer vial. Here you see ascorbic acid that I use for IV therapy in my office. Here's EDTA, okay? So here's the same, vial. And what I did was I just took a drop of the Pfizer vial contents and I mixed them with EDTA and ascorbic acid. And in 10 minutes, it deactivated the vaccine, all nanotechnology, all nanorobots, and all microchips. It just absolutely renders them completely, it's like a nuclear bomb was dropped. So this confirms what I've been saying now for 3 years, and I've been, reversing people's brain fog and heart palpitations and all this stuff. I use it IV, and people can use it orally. Here is a higher magnification. You see that nothing's moving, nothing's blinking. In 10 minutes, this is just, done and over with.
This is amazing. Right. So right out of the patent. I love that. And so EDTA can be used both orally and IV, and the same for vitamin C. Is that right?
Yes, absolutely. So EDTA is a metal chelator, has been used for a long time to chelate metals. Here, by the way, is a higher magnification, 2,000 times. You see that none of the bots are moving. And so you can do liposomal EDTA, for example. You can do it rectally. Here you can see a urine metals test that I did in my office, okay? This patient got 1,500 milligrams of EDTA. And look at aluminum came out, antimony, barium, those are being sprayed via geoengineering, cadmium, here's lead and gadolinium, here's nickel, palladium, platinum. So these, for example, gadolinium is also lanthanide. EDTA binds to all of the lanthanides, all of these undisclosed metals that were in the shots, including, by the way, the Argentinian group found uranium. And you can see here, I'm pulling out uranium, I'm pulling out tungsten, all of them. So when you pull out the metals, the semiconductor technology cannot work because it's the antenna and it's the start signal for the polymers.
Right.
And so here, this is clinically, here's somebody who comes to me, long COVID, totally fatigued, has all these polymers, the blood is in extreme distress, the cells are not normal, and so they come for 3 days. I do IV therapy. This is 3 days later with EDTA, vitamin C alternating. I just cleaned up their blood and their symptoms are gone.
Phenomenal. Phenomenal.
So,
wow. Yeah, go ahead.
So I just wanted to show that we also found these microchips in dental anesthetics, okay? And that the University of Colorado Boulder now has confirmed that graphene was in articaine with epinephrine. I'm working with a biological dentist together on this stuff. So, but it's just the same microchips are in dental anesthetics. And again, this also works. And here I show my research on the dental anesthetics, how they're building the microchips, they're building extreme elaborate structures and microelectronics. And again, I mix it with EDTA and vitamin C and it annihilates it. This is the last couple slides show you that the wildlife is also affected. This is venison. I've looked at beef. I've looked at all kinds of animals. I went to the grocery store, bought meat, and the same filaments are in the beef. They're in the wildlife. This is a wild deer. You can see these microelectronics and quantum dot technology here, the same filaments in their blood. This is a wild squirrel. Willie sent me this. This is their blood. And you see that from the geoengineering, there's a complete overlap. This is something to talk about in a different episode, but I just wanted to show you, and in my book, I will show exactly all of the correlation between, the geoengineering and what's happening to our entire biosphere. The transhumanist technocrats, they want to digitize all life on Earth.
Thank you so much. Yes, we're now doing a monthly segment with Dr. Dane Wigington. I think you've mentioned him about geoengineering to make people more aware of this. And I hope that you will be able to have you back when your book is out. It would be great to talk about what's in your book. Well, you've given us quite the tour through this issue of what you're seeing in the vials and what you're seeing in the blood, and that it's pervasive, right? It's not just vaccinated people, it's all people. And now you're suggesting that it's all animals, or a lot of animals as well. Anything you'd like to tell us in closing? We've actually, we've gone the whole half hour. Anything you'd like to say?
I just want to say, that people don't have to be afraid of this topic anymore, because there are solutions. But if you don't pay attention, you don't do anything, then you're not treating the right thing. So I just want to encourage people to look further. And there's a lot more research on my Substack. The book will be coming out, and don't just dismiss it. There's a lot of disinformation out there. This is real. I've seen it with my own eyes. I've documented it now for 3 years.
What is your Substack, Anna?
Anna Michalcza, MD, PhD.substack.com.
All right. Well, thank you so much for joining us and for illuminating this very serious issue for us. And we look forward to having you again.
Thank you so much for having me.